Hello, I'm Stan and I welcome you here today to the Brain Broadcast Show on XN the Extra Special News Network. Today we have a special segment for you that will dive into the exciting world of smuggling. I'm here with Armando, a world-renowned ex-smuggler. However, Armando was no ordinary smuggler. Armando dealt strictly in the smuggling of objects into cells. So Armando, the whole world wants to know, how do you get the materials that you need into and out of cells? That is a great question, Rick, but to be honest, that question is not an easy one to answer. You see, I use many methods to get things such as proteins and glucose into and out of cells. Tell me more, Armando. Well, the two methods that I used were active and passive forms of transport. Passive forms of transport were incredibly easy because they required no energy at all. Active forms of transport were more difficult because they actually required energy. My personal favorite form of passive transport was diffusion. Diffusion is the transport of materials from an area of high concentration to an area of low concentration. This was particularly useful when I needed to get things out of the cell such as carbon dioxide when the cell had too much of it. Another unique subcategory of active transport that I saved for special occasions is osmosis, which is specifically the transport of water through a semi-permeable membrane. That is very interesting, but could you elaborate on the other form of transportation that actually did require energy? It would be my pleasure. The other form of transport, active transport, required energy, and I used it in cases when I needed to move my supplies from an area of low concentration to an area of high concentration. This was particularly useful when I was being paid to remove excess materials out of the cell, to areas that already had a lot of this excess material. Intriguing? What were some challenges you faced? Some challenges that I faced were transporting large loads. However, I soon found a solution for this. Exo and endocytosis. You see, endocytosis is the transportation of large quantities of important materials into a cell. This process occurs when a cell wraps its membrane around a grouping of important materials, then absorbs this grouping into itself. After the cell has taken everything useful from this grouping, it moves it back to the outside of the cell. Then through the process of exocytosis, the cell wall unwraps from around the grouping and expels all the built-up waste at one time. If you are having trouble picturing this, try visualizing a dump truck moving huge portions of matter at one time. That sounds spectacular. Oh, I assure you, it most certainly is. Another thing that I would like to tell you about is my competition. You see, inside the cell, I'm not the only person in the business of transporting materials. There are also others who do what I did, but not as efficiently. They tended to use these interesting things called transport proteins. Transport proteins are proteins embedded in the cell wall that act as toll booths. These toll booths allow larger particles into and out of the cell. These transport proteins play a role in both active and passive transport. In passive transport, they are used exclusively in the process of facilitated diffusion. Facilitated diffusion is almost exactly like regular diffusion except for the fact that it travels through and uses transport proteins. These proteins are used in active transport as well in cases such as a sodium-potassium pump, which transports ions from areas of low to high concentration. This particular process is known for its role in keeping ions and electrolytes in balance. Wow! So far this interview has been eye-opening, and I have learned so much. But I'm going to have to segue into a different topic. Is that okay? Perfectly fine with me. Okay, in that case, can I ask you to discuss with me a more political topic? Fine by me. From a smuggler's point of view, how do you think the fact that our cells, that we were sworn to protect, are now submerged in a hypotonic solution? What effects do you think this might have? Well, honestly, Stan, it does not look good. If a cell is exposed to an hypotonic solution, bad things start to happen. First, water, being a universal solvent, rushes from the solution to the cell, which has a surplus of solvents. This incoming amount of water causes the cell to become hypertonic and potentially explode. So, unless we can get a handle on things, we are not going to be around much longer. However, it won't help if we try to get the cells into a hypertonic solution either. If a cell is exposed to a hypertonic solution, many problems can occur. For starters, a hypertonic solution has an excessive solutes. So the water in the cells will be attracted to this and leave the cells. This mass exodus of water from the cell causes it to become hypotonic, which is not better than hypertonic. Take it from a smuggler with experience: if our leaders don't start cooperating and eating healthier, we are not going to survive. The only way to save our cell is to get it into an isotonic solution and allow it to naturally balance out and reach dynamic equilibrium. I'm sorry, ladies and gentlemen, but that is all the time we have for today. Thank you so much, Armando, for your well-informed opinions. It was really refreshing to talk to a person who has really experienced these things firsthand. This is Stan signing off. I'll see you next week. Goodbye, everyone. Thanks for having me.